Good day, everyone. I am Rosemilin C. Pelenio, your reporter for this topic. So, we are now in Chapter 9, which is about signaling at the cell surface. So, under the chapter, I have here two subtopics, which is signal methodologies and then cell signal receptors. I am going to read first our learning objectives. So at the end of the discussion, learners are able to examine the basic principles of how cells communicate with one another, explain the mechanisms by to how cell-to-cell signaling works, and then evaluate the importance of understanding the different kinds of short and long-range signaling that happen in our bodies. So before we move on with the discussion, I have here first a trivia. So did you know that at this very moment, your cells are sending and receiving millions of messages in the form of chemical signaling molecules? There are two kinds of communication in the world of living cells. So communication between cells is called intercellular signaling, and communication within a cell is called intracellular signaling. Chemical signals are released by signaling cells in the form of a small, usually volatile or soluble molecules called ligands. A ligand is a molecule that binds another specific molecule, in some cases delivering a signal in the process. So ligands interact with proteins in target cells, which are cells that are affected by chemical signals. These proteins are also called receptors. So presented here is a diagram indicating how the cells typically communicate using chemical signals. So these chemical signals, which are proteins or other molecules produced by a sending cell, are often secreted from the cell and released into the extracellular space. There, they can float like messages in a bottle over to neighboring cells. So not all cells can hear a particular chemical message. In order to detect signal, a neighbor cell must have the right receptor for that signal. When a signaling molecule binds to its receptor, it alters the shape or activity of the receptor, triggering a change inside of the cell. Signaling molecules are often called ligands, a general term for molecules that bind specially to other molecules such as receptors. So here in the illustration, it implies that the chemical messengers relay the signal. So the message carried by a ligand is often relayed through a chain of chemical messengers inside the cell. Ultimately, it leads to a change in the cell, such as alteration in the activity of a gene or even the induction of a whole process, such as cell division. Thus, the original intercellular signal is converted into an intracellular signal that triggers a response. We are now in forms of signaling, so the cell-to-cell signaling involves the transmission of a signal from a sending cell to a receiving cell. However, not all sending and receiving cells are next-door neighbors, nor do all cell pairs exchange signals in the same way. So there are four basic categories of chemical signaling found in multicellular organisms, which includes the paracrine signaling, autocrine signaling, endocrine signaling, and signaling by direct contact. The main difference between the different categories of signaling is the distance that the signal travels through the organism to reach the target cell. The first basic category of chemical signaling is the paracrine signaling. Often cells that are near one another communicate through the release of chemical messengers, ligands that can diffuse through the space between the cells. This type of signaling, in which cells communicate over relatively short distances, is known as the paracrine signaling. Paracrine signaling allows cells to locally coordinate activities with their neighbors. So one specific example for this category of chemical signaling would be the spinal cord development. An example of a paracrine signaling is what we call the synaptic signaling. So it is in which the nerve cells transmit signals. This process is named for the synapse, the junction between two nerve cells where signal transmission occurs. When the sending neuron fires, an electrical impulse moves rapidly through the cell, traveling down a long fiber-like extension called an axon. When the impulse reaches the synapse, it triggers the release of ligands, called neurotransmitters, which quickly cross the small gap between the nerve cells. The neurotransmitters that are released into the chemical synapse are quickly degraded or taken back up by the sending cell. This resets the system so the synapse is prepared to respond quickly to the next signal. So what happens here during the paracrine signaling is that a cell targets a nearby cell. This mainly occurs between local cells as it allows cells to communicate with each other by releasing signal molecules, while during the autocrine, a cell targets itself. So, it is a type of cell communication occurs when the cell secretes a substance that is capable of provoking a reaction in itself. So again, in autocrine signaling, a cell signals to itself, releasing a ligand that binds to receptors on its own surface. This may seem like an odd thing for a cell to do, but autocrine signaling plays an important role in many processes. For instance, autocrine signaling is important during development, helping cells take on and reinforce their correct identities. From a medical standpoint, autocrine signaling is important in cancer and is thought to play a key role in metastasis, the spread of cancer from its original site to other parts of the body. The third category, which is the endocrine signaling. When cells need to transmit signals over long distances, they often use the circulatory system as a distribution network for the messages they send. In long-distance endocrine signaling, signals are produced by specialized cells and released into the bloodstream, which carries them to target cells in distant parts of the body. So here, the signals that are produced in one part of the body and travel through the circulation to reach faraway targets are known as hormones. So during the endocrine signaling, a cell is said to be the targets in the distant cell through the bloodstream. So again, from the signaling cell, going to the bloodstream, up until it reaches the target cell. The last category for the chemical signaling, which is the signaling by direct contact. So gap junctions in animals and plasmodesmata in plants are tiny channels that directly connect neighboring cells. These water-filled channels allow small signaling molecules called the intracellular mediators to diffuse between the two cells. Small molecules and ions are capable to move between cells, but large molecules like proteins and DNA cannot fit through the channels without special assistance. In this category of chemical signaling, a cell targets a cell connected by gap junctions. The transfer of signaling molecules transmits the current state of one cell to its neighbor. This allows a group of cells to coordinate the response to a signal that only one of them may have received. Moving on with the types of receptors. So what comes in your mind when you hear the word receptors? So it could be it is composed of protein, or even it is a chemical structure. So yes, receptors are protein molecules in the target cell or in surface that bind ligand. There are two types of receptors, namely the internal receptors and the cell surface receptors. So the first type would be the internal receptors. So it is also known as intracellular or cytoplasmic receptors. These are found in the cytoplasm of the cell and respond to hydrophobic ligand molecules that are able to travel across the plasma membrane. So once inside the cell, many of these molecules bind to proteins that act as regulators of mRNA synthesis to mediate gene expression. Gene expression is the cellular process of transforming the information in a cell's DNA into a sequence of amino acids, which ultimately forms a protein. When the ligand binds to the internal receptor, a conformational change is triggered that exposes a DNA binding site on the protein. The ligand receptor complex moves into the nucleus, then binds to specific regulatory regions of the chromosomal DNA and promotes the initiation of transcription. Transcription is a process of copying the information in a cell's DNA into a special form of RNA called messenger RNA. The cell uses information in the mRNA, which moves out into the cytoplasm and associates with ribosomes to link specific amino acids in the correct order, producing a protein. The second type of receptor is called the cell surface receptors, which are also called as transmembrane receptors. So these are membrane-anchored proteins that bind to external ligand molecules. This type of receptor spans the plasma membrane and performs signal transduction, in which an extracellular signal is converted into an intercellular signal. Ligands that interact with cell surface receptors do not have to enter the cell that they affect. Cell surface receptors are also called cell-specific proteins or markers because they are specific to individual cell types. So we have here the types of cell surface receptors. First would be the ion channel linked receptors, G-protein linked receptors, and the enzyme linked receptors. So ion channel link receptors bind a ligand and open a channel through the membrane that allows specific ions to pass through. To form a channel, this type of cell surface receptor has an extensive membrane spanning region. In order to interact with the phospholipid fatty acid tails that form the center of the plasma membrane, many of the amino acids in the membrane spanning region are hydrophobic in nature. Another type of receptor is mainly known as the G-protein link receptors, which bind a ligand and activate a membrane protein called G-protein. The activated protein then interacts with either an ion channel or an enzyme in the membrane. All G-protein link receptors have 7 transmembrane domains, but each receptor has its own specific extracellular domain and G-protein binding site. Cell signaling using G-protein link receptors occurs as cyclic series of events before the ligand binds, then active G-protein can bind to a newly revealed site on the receptor specific for its binding. So the last type of receptor is mainly known as the enzyme link receptors. So these are cell surface receptors with intracellular domains that are associated with an enzyme. In some cases, the intracellular domain of the receptor itself is an enzyme. Other enzyme-linked receptors have a small intracellular domain that interacts directly with an enzyme. This receptor is normally have a large extracellular and intracellular domains, but the membrane spanning region consists of a single alpha helical region of the peptide strand. When a ligand binds to the extracellular domain, a signal is transferred into the membrane, activating the enzyme. Activation of the enzyme sets off a chain of events within the cell that eventually leads to a response. Okay, class, that ends my presentation. Thank you for listening.